this one microbe can cause all these different conditions. It has to do with their valence factors. When we look at Streptococcus pyogenes, we can see that it's got several different mechanisms that it employs that help it avoid some of the host defense mechanisms. One of the things it does is it produces a C5 peptidase that interferes with complement. Remember, C5 complement here is complement that attracts macrophages to the area. So if you release an enzyme that breaks down that complement, it's going to slow down the movement of macrophages to the area. Gives you a little bit of chance to multiply. This microbe produces a capsule. And in this capsule, hyaluronic acid this is an acid molecule that we also possess. And so it's one of our molecules, and so our microbes don't necessarily look for it to destroy it. And so it provides sort of a cloaking, if you will, to the microbe. Our cells have hyaluronic acid. Our immune cells wouldn't necessarily recognize it. So this capsule with this molecule is cloaked from our immune system. So it's protected in that sense. On the surface, there's a protein, an M protein, that helps degrade complement C3. Now, if you remember what C3 does, this is a complement that binds and abscindicizes microbes. So it kind of serves to flag those microbes. What you need to recognize, all of this, and I'm not saying every strain of Streptococcus necessarily does all of these things, but the fact that you're able to do these things initially gives you a chance to replicate before the immune system has a chance to attack you. And that's one of the ways that microbes are able to cause infection, is if they can increase their replication, or if they can multiply before your immune system has a chance to respond, then they have a chance to establish infection. If your immune response is such that these guys don't get a chance to replicate, then you're not going to see an infection established. So you can see that your microbes are working to avoid that immune system. Protein F, also found on the surface of the cell. This mediates attachment. So this is going to allow the cell then to bind to the right epithelial cells. And you have to also recognize that as well. When we're talking about microbes and the cells that they infect, generally they're infecting very specific cells. Cells that they have a protein that allows them to bind to the surface receptor of those cells. Same with toxins. Anytime we're talking about a toxin that's being spread to the system, that toxin is binding to only specific cells that have a receptor for it. If the cells lack the receptor, then the toxin won't bind to it. So sometimes you can see a toxin that targets kidney cells or targets liver cells because it has, those cells have a receptor that toxin can bind to. Protein G. This is a molecule, again, found on the surface of the cell that binds to antibodies, but it binds to the Fc region. So you understand the significance of that now? Here we go, our bacteria, and instead of the antibodies binding like this, so that now the macrophage can come along and recognize the FC and devour. Instead, the antibodies are binding like this. So the macrophage isn't going to recognize that. It's not going to devour that. And again, the bacterium is kind of cloaked in this way. SPEs, streptococcal pyrogenic exotoxins. So we're talking about any number of molecules that the microbe might secrete that could have a toxic effect. Scarlet fever is caused by an exotoxin. Necrotizing fasciitis is caused by an exotoxin. These toxins, they serve as super antigens. And if you remember what that means then, it means that they turn on way too many T cells. So normally 1 T cell out of 100 might get turned on. Super antigen might turn on 5 or 10 out of 100. And so now you've got all these T cells all secreting their cytokines. And so that increase in cytokines can cause damage to cells. Cytokines themselves can do the damage. Streptolysin molecule O, streptolysin molecule S, both of these proteins are responsible for beta hemolysis. Beta hemolysis is then destruction of red blood cells. And so one of the ways that we can test for this microbe is to streak it onto a blood agar and look for a clearing around the colonies. If there's a clearing around the colonies, that means that the microbe is secreting the toxin that's causing the destruction of the red blood cells. And so that's the reason we see the clearing. Other enzymes. DNases, proteases, hyaluronidases, all of these things can serve to allow for the infection to catch hold, if you will, and spread. Fortunately, we can treat Streptococcus pyogenes with antibiotics. Penicillin is effective, erythromycin is effective. Again, you'll have probably a 10-day course on that treatment. Any questions about this microbe? That's all Streptococcus pyogenes. I would expect that you would be able to identify any one of these diseases with that microbe or any one of those virulence factors with that microbe. So in a matching kind of situation.
So all those infections are just where the microbe goes? Is that why they're...
Exactly, where the microbe goes or what it does. Now, not all strains of Streptococcus are going to produce all the toxins that can be produced. So some of them may cause scarlet fever, some of them may not cause scarlet fever, depending on the toxin that they're able to produce. And of course, if you're not infected with a virus, you would be causing the condition too. So these are all conditions caused by that microbe, but I'm not saying one strain causes all of those conditions.
And they have different vehicles of transmission. I mean, they can be contact, they can be...
Most of this is by droplet nuclei, close contact. In some cases where we're talking about skin conditions, you would have to have direct contact to those places, yes.
Earlier you said that they're respiratory because they have to be inhaled. Is that the case with all these diseases? Or
Well,
these
no.
are all just related to the streptococcus?
They're all related to Streptococcus, and most of them do require inhalation, but of course this one doesn't,
Right.
this one didn't, and this one doesn't. And so we put Streptococcus with the respiratory diseases because it's found deep in the respiratory tract. But you can see it can cause other conditions that aren't necessarily respiratory. And we'll see also that there are some conditions that we classify as skin conditions that are spread via the respiratory. So take that for what it is. Any questions here? So let's continue to talk about diseases of the upper respiratory system. Diphtheria. A disease that you're immunized for very early in life with the DPT vaccine, the diphtheria pertussis tetanus vaccine. This one is caused, diphtheria is caused by microcorinabacteria diphtheria. It's a toxin-mediated disease. So that means that the microbe is releasing a toxin that causes the disease, and in this case, we're talking about a condition in which the microbe has been lysogenized again. So that means if the virus isn't there, the microbe isn't producing the toxin.
So what you're saying is the virus has infected the microbe.
Yes. And it's the presence of the virus that causes the production of the toxin. So with this, you get a gray, white mass, if you will, grayish white mass of clotted cells, red blood cells, epithelial cells, mucus and stuff that grow along the respiratory tract right there on those lining. And what can happen is it can break free and it can cause suffocation to occur. The other thing about the disease here is that when the toxin goes to work, it's an AB toxin. Excuse me, the B part identifies specific organs, specific cells that it infects. And this is what I was referring to with specificity here. This particular microbe, or the toxin I should say, is going to target kidney, liver cells, and the A chain actually interferes with protein synthesis. Interferes with transfer RNA so that you can't get protein synthesis and then the cell dies. Can be immunized with it, as I said. We use a toxoid for immunization. So this is the toxin that's been changed so that only the antigenic epitope is there. The virulent part has been removed from the toxin. We can also use antiserum for treatment if you're already infected. So this is, something that a serum that's already been produced by an animal or a person that's got antibodies in it for that particular toxin. A couple of other upper respiratory conditions we'll just mention here. Pink eye, earaches, sinus infections can be caused by any number of microbes. Streptococcus pneumonias, Haemophilus influenza, Bacillus strains, Pseudomonas strains, any number of microbes can cause conditions like pink eye, earaches, and sinus infections. So these are, the ones that I've mentioned here are bacterial infections of the upper respiratory system. One of the ways that you might want to organize your data here is to keep track of where the microbes are infecting upper or lower, and whether we're talking about bacterial versus viral. So the next group here I want to talk about are the viral infections of the respiratory system. And the main one that we're going to mention here the common cold. The average person is going to have upper viral infection every 2 to 5 years. Excuse me. The average person is going to have 2 to 5 upper respiratory viral infections per year. So over your lifetime, you're going to have about 200 colds, give or take, depending on how healthy you are and what you expose yourself to. This common cold, most of us will have 2 to 3 colds a year. We will develop an immunity to that particular virus, but it's going to be short-lived. It's only going to last about 3 years. It's very short-lived immunity. About 30 to 50% of common colds are caused by the rhinovirus. But there are a whole number of others that can cause the conditions that we know as a common cold. The virus attaches to specific receptors on epithelial cells. It multiplies there, goes on and infects other cells, destroys the ciliary motion so that you develop that cough to get